Good morning and Jai Hind children. I welcome you all in today's live class of biology. Children, in previous class, I have revised some of the topics of the chapter nutrition in plants. Is it? Now today I am going to demonstrate the activities of the chapter which is given in the syllabus. So the activity number 1 is to show that light is necessary for photosynthesis. Okay, so first of all, I'm going to demonstrate this activity number 1 to show that light is necessary for photosynthesis. Now, children, today I'm going to demonstrate you the activity number 1 of biology, and the aim or objective of this activity is to show that light is necessary for photosynthesis. So, children, for this activity, we require some materials like we require a potted plant. So, here I have a potted plant. We require some black paper strip to cover the half part of leaf. So, here I have covered the leaves with the black paper strip. I have applied the paper strip and with the help of paper clip, and will keep this setup in the sunlight for about 2 hours for photosynthesis. Now, after that, we also require some more material for this activity. Like we require burner. I have the spirit lamp here. We require tripod stand. We require wire gauge. We require beaker, water. We require also the ethanol, ethyl alcohol solution, and we require iodine solution to taste for star. Now, the procedure is after 2 hours, when we'll bring our potted plants back. So first of all, we will pluck the leaf which we have covered and removed that paper which we have applied there, the black paper which we have applied there. This was done because the covered part will not get the light as it's get covered with black color, and uncovered part have get the sufficient light. After removal of this paper, we will light our burner and put the wire gauge on it and a beaker with water, little water, and the leaf which we have taken out and gets uncovered from that, we will put it in this water and allow them for boiling. So after 4 or 5 minutes, when the leaf get boiled, the cells of the leaf gets die and it's get loose. After that, we will take a test tube, and in this test tube, we will take some ethyl alcohol or ethanol. So about half of the test tube, ethanol I have taken here in this test tube, and will transfer this leaf which are boiling from last 2-3 minutes in this test tube which contains the ethanol. So with the help of a glass rod or with the help of the forceps, we will transfer it and we'll put it here in this test tube. Now the leaf is get kept here in this test tube. Now we will put this test tube in which we have the boiled leaf and ethanol in this water which is hot, and allow the ethanol to boil. But our water should not get boiled, as the boiling point of water and ethanol is different. The boiling point of ethanol is less than water, so the ethanol will start boiling first before the water. So we have to take care that our water will not allow to boil, only the ethanol is allowed to boil. As ethanol will start boiling, so we'll observe that the ethyl alcohol, the solution gets greenish because the leaf will lose its chlorophyll, and the solution will get green in color, and the leaf will becomes colorless. It lose its color. After that, we will take a petri dish and spread that leaf in this petri dish, which we are boiling here in this test tube. So with the help of this rod, I will take out the leaf. And the iodine solution which we have taken, will put some of the iodine solution with the help of dropper in this petri dish where I have kept the leaf. Now you will observe here the part which was covered by black spray, it's remained colorless, whereas the part which was not covered, it's get blackish or dark brown in color. So here I have the leaf. You can see very clearly that some of the part of the leaf gets dark brown in color, whereas the uncovered part is remain colorless. So this indicates that for photosynthesis reaction, light is necessary. Without light, the photosynthesis will not occur, and as a result, the starch will not form. So I hope that you understood this. So, I hope that you understood the activity that light is necessary for photosynthesis. Is it, children? Okay, very good. Now, we have one more activity in this chapter, the activity number 2. And these both activities are there in the syllabus, so you need to write these activities in your activity notebook. Alright, children? And the aim or objective of activity number 2 is to show that carbon dioxide is necessary for photosynthesis. Okay, so now I'm going to demonstrate you that carbon dioxide is necessary for. So, children, today we will demonstrate the activity number 2 of the biology syllabus, and the objective of the activity is to show that carbon dioxide is necessary for photosynthesis. For this activity, we require a potted plant. So here I have a potted plant which have many branches. We will take a small test tube and tie a thread. Here I have a small test tube. I have tied a thread in this test tube and put some potassium hydroxide solution. So I'm going to put some potassium hydroxide solution in it. Now tie this test tube with one of the branch of this potted plant. So one of the branch I'm going to tie this test tube. Any branch you can select which have the leaves. Now I have tied here in this branch, and now this branch which I have this potassium hydroxide, I am going to cover with a polythene bag, and that polythene bag should be transparent. And I will tie it tightly with a thread so the air is not allowed to enter in it. Some of the branches are uncovered. Now, put this setup in the sunlight for about 2 to 3 hours. When the setup was kept in the sunlight, the photosynthesis process will occur. As the polythene bag was transparent, so sunlight will also enter in it. But inside that polythene bag, air is not allowed to enter because I have tied it completely and tightly so that air is not allowed to move inside it. Only the light is allowed to. Now, after 2 to 3 hours, I will take out the potted plant bag and remove this polythene bag, and I will also remove this potassium hydroxide solution which I have tied here. Now, I will pluck the two leaves from this plant. The one leaf from the covered branch of the plant and the one leaf from the uncovered branch of the plant. So, one covered leaf and one uncovered leaf. Both the leaf I will put in the beaker. So here I have a beaker with water. Now both the leaves I will keep it here and allow them to boil. So now I'm going to light the candle, the burner, put the wire gauge on it, and then the beaker with leaf, and allow them to boil for about 3 to 4 minutes. So after 2 to 4 minutes, the leaf gets loose because the cells of the leaf gets killed. And after that, we'll transfer these leaves in a test tube with have the ethyl alcohol solution or ethanol solution. So here I have a test tube and I'm going to put the ethanol solution in it, and it should be about half of the test tube, and with the help of the glass rod I will transfer these leaves in this test tube. So I have kept the leaves in this test tube. Now I will keep another beaker with water and shift this test tube in this beaker of water and again heat it. And this time we will take care that our only ethanol solution is allowed to boil, not the water. As the ethanol is having less boiling point than water, so it will start boiling first, that before the water. So we have to take care that only the ethanol is going to boil. So after few minutes, we'll observe that the ethanol is going to boil and the green color of the leaves are going to remove and the leaves become colorless. Now we'll transfer it and shift these leaves in the petri dishes. So with the help of the same glass rod, I'll transfer the leaves in this petri dish and spread them properly. So here I have both the leaves which get colorless. Now I will pour iodine solution in both the petri dishes. Here I have the iodine solution. With the help of dropper, I will pour this iodine solution in both the petri dishes. And observe that the change of color in one of the leaf which was covered and the leaf which was not covered, its color gets changed. That gets blue black, a dark brown in color. So you can see here that the leaf which was not covered gets dark brown in color, while the leaf which was covered, it's remained the same, colorless. So this difference in the leaves indicates that the branch which was covered, it not got the carbon dioxide, and whatever the carbon dioxide was present in the bag get absorbed by potassium hydroxide. So in the absence of carbon dioxide, the process of photosynthesis not occurred there, and as a result starch not formed, and there is no change in the color of the leaf. Whereas the leaf which was not covered, it got the carbon dioxide in sufficient amount, photosynthesis occurred there, starch formed, and as a result, the color of the leaf gets changed blue. So children, I hope that you understood this activity. So children, I hope that you understood the second activity. Okay, that to show carbon dioxide is necessary for photosynthesis. Alright children, so write these both activities in your activity notebook. I have already provided you the PDF of these activities. Okay, now come to the rest of the topics of the chapter. So I'm going to revise those topics of the chapter, and today the topic that I will revise is the synthesis of food other than carbohydrate by plants. Children, as you know that when the photosynthesis occur in the plant, they prepare the food, and that food is mostly the carbohydrate or glucose. Okay, but this carbohydrate that plants synthesize during the photosynthesis are made up of carbon, hydrogen, and oxygen. Is it? They are made up of, yes, the three elements. The first element is carbon. Second element is, yes, hydrogen. And the third element is oxygen. Alright children. Now these are used to synthesize other components of food such as proteins and fats. Get it? And the proteins that contains nitrogen along with these three elements, that carbon, hydrogen and oxygen, it also contains nitrogen. And the plants cannot absorb nitrogen directly from the air, and they get this nitrogen in two different ways. Got it, children? So you can say that there are two ways by which plants absorb the nitrogen, and the first way is, yes, from the soil, because the soil contains certain bacteria which are called rhizobium. So the name of bacteria is rhizobium bacteria. It can convert the atmospheric nitrogen into water. Okay, so this rhizobium bacteria can convert the gaseous nitrogen or the atmospheric nitrogen into the water soluble compounds. And these water soluble compounds of nitrogen absorbed by the plants. Got it? Understood, children? And the plant fulfill the requirement of nitrogen to form the proteins. So this is the first way by which plants take the nitrogen, and the second way is that the addition of fertilizers which are rich in nitrogen. So you must have seen that farmers add fertilizers which are rich in nitrogen to their soil. Okay, and these are absorbed by the plants. So the two ways by which plants get the nitrogen, the first way by the rhizobium bacteria and the second way by fertilizers. Alright, children? Now, this is all about the autotrophic mode of nutrition in plants. Okay? Now, I'm going to discuss the second mode of nutrition. And that second mode of nutrition is what, children? Yes, it is heterotrophic nutrition in plants. Get it? So, come to the second mode of nutrition. That is heterotrophic mode of nutrition. Heterotrophic nutrition in plants. As you know very well, the meaning of heterotrophic nutrition, the mode of nutrition in which the organism is not capable to synthesize the food by themselves. Okay, so they depend on other organism, other green plants. Got it then? So there are different types of heterotrophic nutrition which are found in the plant. And the first kind of heterotrophic nutrition which is found in the plant is parasitic plants. Parasitic plants, because the some non-green plants, they live in or on the other living organism and derive their food from them. Got it? And such plants are called parasites. Understood, children? And the example of some parasite plants are dodder, cuscuta, etc. So these are the plants, but they are not green in color. So that is why they cannot do the photosynthesis, and they depend on other living organism to derive their food. And this is why they are known as parasitic plants. Okay, and the plants from which a parasite gets its food is called a host. Okay, these plants, non-green plants, are known as parasites, and the other green plants from which it get their nutrition is known as host. And all the parasitic plants have modified roots which are called hostoria. Get it, children? Hostoria is the kind of roots which are found in parasitic plants. It is just like the needle shaped, by which it can easily injected in the, yes, body of the other green plants and suck the nutrients. Get it? So such hostoria penetrates the host plants and suck the food, water and minerals and fulfill the requirement of the nutrition for their body. Get it, children? So this is the one of the kind of heterotrophic nutrition, that is parasitic plants. Now the next one is the insectivorous plants. But before that, I want to explain that the parasitic plants are of two types. Get it, children? The first one is total parasite, and the second one is partial parasite. The total parasites are those plants, are those parasitic plants which derive all of its nutritional requirements, all of the nutrients from the host plant. Get it? Like dodder, rafflesia. These are the names of some parasitic plants which are total parasite because they completely dependent on host for their food, water, minerals and all. Get it? And that is why they are called total parasites. The second one is the partial parasite. As the name indicate, partial means they are not completely dependent on host. They dependent partially. Okay, so these parasite plants derive some of its nutritional requirements from host plant. Understood? Like mistletoe. Mistletoe is a parasitic plant which derive only some part of the nutrition like minerals and water. Whereas it can do the photosynthesis because it is green in color. Got it, children? So that is why it is known as partial parasite. Okay, so there are two kind of parasites, that is total parasite and partial parasite. All right. Now come to the second kind of heterotrophic nutrition, and that is insectivorous plant. Insectivorous plants catches insects by ingenious method and consume them to fulfill their nutritional requirements. Okay, children, as they feed on insects, so that is why they are termed as insectivorous plants. Okay, and the example of such insectivorous plants are pitcher plant, Venus flytrap. These are some names of insectivorous plant who used to catch the insects, kill them, and suck the nutrients. Got it? And the cells of these plants secrete the digestive juices to digest the insects after trapping them. And these plants grow in the soil that is not so rich. That is why they need to kill the insects to fulfill the nutritional requirement, as the soil is not so rich with nutrient. Okay, and they use the nutrition obtained from insects to supplement the food they prepare by photosynthesis. So you can say that the insectivorous plants are green in color. They can do the photosynthesis, but as the soil is not so rich with nutrients, so to fulfill the requirement of those nutrients, they used to kill the insects and suck those nutrients and use those nutrients to prepare their food. Got it, children? Now, after the insectivorous, the next kind of heterotrophic nutrition is saprotrophic nutrition. So, children, the third kind of heterotrophic nutrition is saprotrophic nutrition. Okay, the method of getting nutrients from dead and decaying matter in the form of a liquid is known as saprotrophic nutrition. Okay, and the fungi and the bacteria use this mode of nutrition and are known as saprotrophs or saprophytes. Got it? This mode of nutrition is known as saprotrophic mode of nutrition, and those organisms are known as saprotrophs or saprophytes. The saprotrophs, they secrete the digestive juices on the dead and decaying matter, which converts the solid matter into a liquid. And when they convert it into liquid, then they absorb the nutrients from this liquid and fulfill the requirement of nutrition for their body. Got it, children? So you must have to remember that the difference in insectivorous plant, parasitic plants and saprotrophic plant. Parasitic plant obtain their nutrients from living organism, the host which is living, whereas insectivorous, they used to kill the insects. Whereas saprotrophic mode of plants, they grow on dead and decaying matter. Okay, and release the digestive juices, digest that dead matter, and absorb the nutrients from that. Got it, children? Now, we have one more kind of heterotrophic nutrition, and that is symbiosis. Children, in symbiosis, the two different kinds of organisms work together for their mutual benefit. Okay, such as mutually beneficial relationship is called symbiosis. So you can say that in symbiosis, both the organism get benefited. Okay, and the example of this symbiosis is lichens. So lichens are one of the example of symbiosis. In lichens, an alga, which is an autotroph because they can do the photosynthesis, and a fungus, which is a saprophyte because they cannot do the photosynthesis. So they both live together. That alga and fungus live together. The fungus supplies water and minerals to the alga, and the alga supplies food to the fungus, and they both get benefited. Alright, children. So such a relationship between alga and a fungus is known as lichens. Okay, and this is the example of which kind of heterotrophic nutrition? Symbiosis mode of nutrition. Alright, children. So these are the different kind of symbiosis, different kind of heterotrophic nutrition. Now, one more thing, children, that you must have to know, that plants which absorb the nutrients from the soil, therefore the amount of nutrients in the soil goes on decreasing as the plants continuously absorb the nutrients, so their level of nutrient get decreased, and in a forest these nutrients get naturally replenished by decaying of dead plants and animals. Okay, so continuously the nutrients get replenished and absorbed by the plants. And on a farm, these nutrients have to be added to the soil in the form of manure and fertilizers. Because in the farm, the dead and decaying matters are not allowed to decompose there more. So that is why replenishment is very, very less. So farmers use to add manures and fertilizers in their farms to maintain the level of nutrients. Okay, and a farmer knows which nutrients are crucial for which crop and adds manure and fertilizers accordingly. Got it? And most of the crops require a lot of nitrogen to make protein. And after the crop is harvested, the soil becomes deficient in nitrogen. Like the, yes, like the legume crops. And the bacteria rhizobium, which already I have discussed, this bacteria can take atmospheric nitrogen and convert it into a soluble form. Okay, and from this soluble form of nitrogen, it can be absorbed by the plants. Got it, children? So this is the way by which the nutrients get replenished in the soil. Understood? So this one all about the revision of the chapter. Understood, children? So I hope that you understood this overall chapter along with the activities. Thank you and have a nice day, children.